Okay, hi, everybody. How are you? I am here again with the next portion from the properties of enzymes. The remaining portion today, I will present you the remaining portion. But before we proceed to our lecture, don't forget subscribing, liking, sharing these videos. Please also put your comments under this here, and let's go. Next portion is this one function of enzymes. We shall talk about function of enzymes. Enzymes help speed up chemical reaction in the human body, and they are essential for respiration, digestion, food, the liver, muscle, nerve function. Every biological process is dependent on biochemical reaction, either catabolic or anabolic reaction. Each cell in the human body contains thousands of enzymes that provide help in facilitating chemical reaction within each cell. You see, if you are asked biochemically, if you ask a biochemist what life is, oh, please, life is nothing but uh, the totality of metabolism. Just it's metabolism. What's metabolism? The total set of reactions, catabolic or anabolic reactions occurring in each cell. Of course, reactions can also occur outside of cells, like the reaction that occurs by extracellular enzymes, the digestive system. Of course, therefore, life is possible because of those biochemical reactions. And for these biochemical reactions to occur in their appropriate speed, the speed that life requires to sustain living, we require the service of catalysts, the service of enzymes, biocatalysts. Therefore, the turnover number of molecule is the number of substrate molecules converted by one mole of one enzyme molecule per second at saturated, fully occupied active states. You see, this is the definition. Turnover number, one mole, make it one mole enzyme per second at saturation point at maximum active, fully occupied. When the reaction reach maximum speed, we can calculate the turnover number. Enzymes are markers of the states of various diseases like myocardial infarction. You see, we mention here turnover number because they are highly efficient. Highly efficient. We require small amount, highly efficient, turning thousands of substrates into products. Speed is what life requires. The other is enzymes are markers of the states of various diseases like myocardial infarction, jaundice, pancreatitis, cancer, neurodegenerative disorders. We have made use of the knowledge in enzymes. For example, if you find an enzyme that is only restricted, an enzyme which naturally is only found in the liver. If you find this enzyme in your circulatory system, which should, by normal condition, which should never come out of the liver cells, if you find it in your circulatory system, you have to suspect your liver for abnormality because it is leaking its enzymes. It is not containing its enzymes properly. There may be some damage because of alcohol or some damage because of some viral infections in your liver. Therefore, pancreas is the same. There cancer also we suspect. We analyze enzymes from different fluids or tissues in the body. We undergo diagnosis, and this can tell us indication for some health abnormalities. Therefore, we use enzymes for diagnostic purpose. That means each enzyme has an active site with a unique shape that speed up metabolic or chemical reaction in our bodies and builds substances in all living things. Of course, building or breaking reactions. Of course, examples of enzymes you can see sucrase, lactase, carbohydrates, lipase, protease. You see, you can see here diagram enzyme function. The substrate binds to the active site, producing enzyme substrate complex reaction. The product is free, and the product new substance is produced. That is breaking reaction. The substrate in this case is sucrose, of course. The enzyme is sucrase, of course. And finally, glucose and fructose. The bond between glucose and fructose broken down by this enzyme, so that the enzyme is free at the end of the reaction. Enzymes perform their function by lowering a reaction's activation energy. Activation energy is the energy required to start a reaction, uh, energy required by the substrate molecules to undergo a change into a product, of course. The lower the activation energy, the faster a reaction happens. We have told this earlier. You can see here this diagram. This diagram, the energy versus reaction course graph, and the substrate is here. In the absence of enzyme, this much amount of activation energy, you see, this much huge amount of energy is required. So, this huge amount of activation energy can be obtained only by very few substrates from their environment. So, very few substrates change into product. If because of activity of catalysts, in this case enzymes, the activation energy is hugely decreased, many of the substrates can easily get from their environment in the form of heat, so that many substrates convert into product per unit time, which means increasing the rate of the reaction. That is important for life to keep on living. Some of enzymes in the body and their functions, it says, you can see here, salivary amylase, the other name is thiolate, secreted by salivary glands. It converts starch into maltose. The other is renin. Renin in the stomach of young calves, of course, converts milk proteins to peptides. The other is pepsin in the stomach, also converts as proteins to peptides also. Gastric amylase, gastric amylase. There is no enzyme produced by stomach, of course, but the amylase that comes from saliva together with food that we swallow. There may be some starch digestion, of course, in the maltose, but not usually mentioned here. As far as your book is concerned here, gastric amylase, I don't know, an enzyme produced by gastric gland cells. Gastric amylase is the amylase that comes together with your food during swallowing from the salivary gland. Gastric lipase, gastric lipase converts butter fat into fatty acid and glycerol, and trypsin from pancreas converts proteins to peptides. It says. There is chymotrypsin. Pancreas converts proteins to peptides. Uh, trypsin, pancreatic lipase converts fat into fatty acid and glycerol. Carboxypeptidases, these are peptide breaking into amino acids. They break down peptides into amino acids. Pancreatic amylase converts starch into maltose. Enterokinase, small intestine, enterokinase activates trypsinogen to trypsin, the inactive form trypsin into trypsinogen. Enterokinase is an enzyme in the wall of the small intestine which sits there and breaks down the inactive of trypsinogen into trypsin. Repsin, small intestine, converts polypeptides to amino acids. Repsin is also produced by pancreas, small intestine. Maltase, small intestine, digests maltose into glucose. Sucrase, small intestine, digests sucrose into glucose and fructose. Lactase, milk sugar is changed by lactase into glucose and galactose. It is enzyme in the brush border of the small intestine. Thank you very much. This is the end of this slide. Please don't forget subscription and liking and sharing my videos. Next time I will come by another presentation on enzymes.